without further ado, I'll hand the mic over to my colleague, Dr. Katie, and Katie will run us through both the case studies and the technology session. Thanks for your time, thanks for attending, and look forward to hearing your thoughts on today's webinar.
Thanks, Chip. I'm really excited to be here today and chat with everyone and just share some of the work that Metabolon has done with some of our collaborators and some of the people that have used us to collect data for their research. And so hopefully everyone can find this a little bit informative and make a little brainstorm for what they're doing in their own research. Just a little roadmap here of where we're going. We'll talk a little bit about metabolomics. I'll present the case study about the AAV treatment for muscular dystrophy. We'll talk about the platform technology and the key deliverables. And then we'll talk about that second case study with T-cell activation and optimization. And then we'll have a Q&A session. And there will be time for questions after the platform and technology discussion as well. So what is metabolomics? Metabolomics is the analysis of all of the small molecules in a system. So that can be sugars, amino acids, lipids, anything really that you think about when you're thinking about some of those traditional biochemical pathways that you learned about. So TCA cycle, glycolysis, beta oxidation, when you're thinking about energy metabolism, redox metabolism, glutathione, those types of molecules. It's really the analysis of all of those small molecules and everything that makes up the small molecules in the system. And this can be representative of different reactions that are happening. So maybe substrates and products, modifications that are happening to some of those small molecules, how some of those molecules are degrading, or some of those proteins even might be degrading. And it can be really informative of, classic biochemical reactions, binding and dissociation, phosphorylation, those types of reactions. And so it's really a good insight, and it's really the closest to the phenotype when we're talking about assessing your samples and your model and your research. Metabolomics, I like to think about it on sort of the central dogma of biology. So when you think about genomics and transcriptomics and proteomics and that hierarchy of how, biological organisms are functioning and how they make genes and then proteins and how those enzymatic reactions are happening, the metabolome is reflective of that and integrates into that because it's really what is happening. So you're able to see what products are being made, what substrates are being used up, what reactions are happening, where some of those metabolites might be building up, and how some of those pathways are going forward or not going forward. And really it's just the integration of all of those reactions all together. So the genome is kind of what could be there. The transcriptomic analysis can tell you, maybe a little bit more about what proteins might be making or being made. Proteins are, they're telling you what reactions might be happening or, what the cells or the organisms might be needing to utilize. But the metabolome is really telling you what chemistry is happening in real time. And it's really that snapshot of the biology and really that closest to the phenotype. So first, I'm going to talk about our first case study here with our AAV treatment for muscular dystrophy. Excuse me. This first case study that I wanted to share with you is from a group out of the Carolinas Medical Center. It focuses on AAV gene therapy for FKRP muscular dystrophy. This is a muscular dystrophy Dystroglycanopathies is a class of muscular dystrophies that can be categorized as either primary, secondary, or tertiary. It's a congenital muscular dystrophy, and it's an autosomal recessive disorder. It's associated with defects in the processing and functionality of the gamma dystroglycan, which is an extracellular peripheral membrane glycoprotein anchored to the cell, and it's really part of the transmembrane glycoprotein. And those primary, secondary, and tertiary categorizations can be classified as the primary is really a defect on the gene that encodes for the dystroglycans. The secondary is a missense or a mutation on the genes participating in the maturation of the glycans. And the tertiary is a disorder in the genes involved in the fabrication or the carbohydrates or the building blocks that make up those dystroglycans. And this specific one, this FKRP gene mutation, is a fukutin-related protein gene mutation that causes a rare form of limb-girdle muscular dystrophies. then that can result in progressive weakness and degeneration of the skeletal muscles, cardiovascular, and neurological defects as well. The molecular pathogenesis of these dystroglycanopathies are not fully understood. The metabolic phenotype has not been characterized in the past, and there's really a lack of biomarkers and diagnostic targets as well for these. Therapeutic interventions are also really challenging to find. Gene replacement therapy is one option, so this AAV treatment is really one option for the treatment for this FKRP muscular dystrophy. And metabolic response, however, though, to AAV treatments have not been fully elucidated. So that was another game of the researchers investigating this. So they really wanted to characterize not only the FKRP muscular dystrophy, but also the effects of the AAV treatment. To assess this and to get at their hypotheses, the researchers utilized a mouse model. They utilized wild-type mice, control mice, labeled here as C57BL6. They also utilized their FKRP phenotypic mice, which is just labeled here as the FKRP mouse. And then they also treated the mice with an AAV serum therapeutic, and that's labeled here as the AAV9 FKRP mice. They had age and sex matched mice, and they monitored the mice for 52 weeks. And then they did metabolomics on the skeletal quadricep muscles to assess the tissue-specific dysregulations in the FKRP deficient mice, and also to assess the AAV interventions at the metabolic level. The global metabolomics analysis of the muscle tissue of each of the different groups of mice showed good coverage across the biochemical superpathways. You can see there we're covering amino acids, peptide metabolism, carbohydrate, cofactor, and vitamins, antibiotics. There are 524 metabolites that were identified in their study, and there were 247 that were significantly different between the control and the FKRP disease mice. There were 187 metabolites that were significantly different between the AAV-treated mice and the FKRP mice. And then there were 99 metabolites that were significantly different between the AAV and then the control mice. We can see on the principal component analysis that we're getting good separation across principal component 1. So you can see here where that good separation is happening, and each one of our treatment groups is clustered nicely together. On PC2, you can see there's a little bit less separation here, but it still aligns with the sex of the mice. So that circles were actually males and the squares were actually the females mice. And you can see that the AAV treatment put the samples really in between that control and that disease state. So once they were treated with the AAV gene therapy, it shifted those mice and their phenotype closer back to a control phenotype. There were a lot of pathways that were changing that were reflected and on the deeper dive when we're looking at pathway analysis and not just looking at the global overall analysis of the metabolomics here. Specifically, protein degradation was one that was significantly different based on the phenotype of the FKRP versus the control and the AAV treatment. Amino acids and metabolites showed big changes between the FKRP and the control group, which was indicative of protein metabolism and catabolism, which was really affected because of this phenotype. So you can see here with even, we're showing acetylated amino acid here, box plots. You can see the differences. The red is the FKRP phenotype, the blue is the control, and the yellow, is the AAV treated mice. And you can see the significant differences between the control and the disease mice, and then how that AAV treated treatment really brought things back to the abundances of those metabolites back closer to the control group and brought it closer to that control phenotype. Acetylated amino acids specifically are seen as products from protein breakdown, and they are significantly elevated in the untreated diseased skeletal muscle, which you can see here, which we were talking about. And also important were extracellular matrix metabolites, which show differences in S-glycosyltryptophan and 5-galactosyl hydroxyl lysine, which were elevated in the FKRP mice. Trans-4-hydroxyproline was decreased in the FKRP mice. And all of those changes taken together really indicate that there were changes in the collagen metabolism and collagen homeostasis, indicating matrix remodeling in the FKRP mice. Another important pathway that was dysregulated was lipid metabolism. Lipid metabolites was another class of metabolite that were significantly different. Fatty acids are critical sources of energy for mitochondrial oxidation and ATP generation. The researchers had hypothesized previously that they would see changes here as lipids are key to skeletal muscle homeostasis. You can see that the majority of plasmalogens, 89%, were significantly elevated in the untreated mice. The FKRP gene therapy was able to reverse the plasmalogen trend and normalize almost all of them. And the FKRP gene treatment overall restored 46% of the lipids and the metabolic perturbations that were occurring there. This indicates that there was potentially improved transport and or oxidation of fatty acids. And again, you can see here the corrective on our fatty acid pathway here. You can see that corrective action where we're seeing, again, the FKRP in the red, the control in the blue, and the AAV treatment in the yellow. And you can see that there were changes there. And after the treatment, they were returned to a more normal phenotype abundance. Methionine and polyamine metabolism were also significantly different. There were reduced levels of sarcosine, glycine, and S-adenyl homocysteine in FKRP mice, which can be reflective of cellular degeneration or regeneration. And the AAV treatment, again, corrected for polyamine metabolite abundances, which could be useful as biomarker useful for biomarkers. Data from this FKRP muscular dystrophy model showed that characteristics associated with the muscular disorder, we're really able to see that although this isn't characterized as a metabolic disorder, the metabolomics showed really strong perturbations in different metabolic pathways and networks. The AAV treatment was able to correct for specific metabolites that could be useful biomarkers of disease or progression of treatment efficacy. 1-methylhistidine and 3-methylhistidine could be utilized as muscle toxicity biomarkers, C-glycosyltryptophan as a biomarker for evaluating longitudinal changes in muscle biology. There were differences in the polyol pathway, which we didn't talk about today, but that could be used as a potential biomarker for clinical risk associated with FKRP-related dystroglycanopathies. And then there were changes in that polyamine metabolite, which could be used as biomarkers of disease progression and efficacy during gene therapy treatment. So really, this study really highlighted the functionality of metabolomics. The researchers were able to gain a deeper understanding of the FKRP-related dystroglycanopathies. They were able to determine the involvement of previously unsuspected pathological pathways, lipid metabolism, that polyamine metabolism. Those were things that weren't in the literature previously. And they were able to identify potential targets for drug discovery and diagnostics. So this study really demonstrates how metabolites can be useful biomarkers of disease progression and also treatment efficacy. I'll just push right along here. We'll talk about Metabolon platform and key technology deliverables. So who is Metabolon? What is Metabolon? So Metabolon was actually founded in 2000. We're located in the RTP area in North Carolina. We have over 200 employees, which includes over 50 PhDs, I think, at this point. We have a lot of bioinformaticians. We have a whole sample prep team. We have a whole curation team. We really have a really strong group of people that are working on our samples and doing the science and helping you with your research. And all of that really makes us think that we are definitely the leader in metabolomics. We've worked on over 10,000 projects. We've worked in all different sample types. Obviously, we work a lot in human health studies, so we do a lot in human matrices like whole blood, plasma, urine, feces, sort of all of those typical matrices that you think about. But we're also really don't shy away from different types of species or different types of models. We've worked in over 100 species. We have plants, dogs, dinosaur bones were listed. We did a study on that a long, long time ago. So we really don't shy away from anything. I've heard some of the craziest things lately is fish mucus. We've worked on intraocular fluid. We really don't shy away from anything. And because we're so willing to work on different matrices, and because metabolomics has such a wide area of applications, we've been cited in people's research over 3,000 times in really high impact journals. We also have different certifications. We have CAP and CLIA, and we're also an ISO 9001 lab. Metabolon really has two arms to what we deliver and what we typically offer to people. We have our traditional global untargeted discovery panel, where that's a really traditional metabolomics approach where you're casting a wide net and you're looking for as many molecules as possible. We also have some different targeted panels as well, ranging from sort of smaller panels up to our complex lipid targeted panel, which has 1,100 lipids on it. And then we have some single analyte assays as well. So it depends on what sort of support you need and what type of questions you're asking, but we think that we can really help you with all of your small molecule analysis. Talking about that global untargeted discovery panel briefly here, we have over 5,400 metabolites in our library, and those are 85 to 90% of those are validated at a tier 1 identification. So that means that on our mass specs, not only are we collecting and we have categorized data for exact mass, retention time, fragmentation pattern, spectral data, we've also run a neat standard on our platform to be able to validate that the metabolite that we're saying is the metabolite is actually that metabolite. So we're really talking about the biochemistry and the biology that's happening, and there's really not a lot of putative IDs or MS2 matches that were included in our library, so you really have a lot of confidence in our data, and it's the highest level of fidelity. Included in that library, we have I skipped a slide. I'm going to go back, sorry. Included in that library, we have good coverage over all of the typical pathways that you think about. So we cover carbohydrate metabolism really nicely, energy metabolism. When we're thinking about those maps that I showed at the beginning, the TCA cycle, glycolysis, beta oxidation, we have really good coverage there, and we have quite a wide we have a big library. Obviously, we have really good coverage that returns quality results for, covering those super pathways really consistently. We also have a lot of xenobiotics in our library, exogenous metabolites. We see a lot of drug metabolites in our library. We also have 20 years of institutional knowledge, like I was discussing, and a lot of that also is related to microbiome. So we have a lot of knowledge around the microbiome as well. There are a few different levels of interpretation and support that you can get from us when you work with Metabolon. So coming standard with any project that you would do with us, you get a heat map of every single metabolite that we detect in your system, represented in that heat map form. So whatever statistics that are needed, t-test, ANOVA, and COVA, all of those pairwise comparisons and changes are represented in that heat map form with abundances up and down, statistically significant at 0.05, at red or green. And it's really nice to look at your data in that way because you're able to, it's categorized into pathways and super pathways. And so you can see really what's happening at that pathway level, and then you can dive into it a little bit deeper at that granular level. Coming standard with all of our deliverables are some basic statistics as well. So like I said, ANOVA and COVA, t-tests, we do PCA plots, hierarchical clustering, random forest analysis, and then whatever other statistics you might need, we can also help you there as well. We also provide line plots or bar plots of each individual metabolite. We include a CSV machine-readable data file. If you want to take your data and work with it in R or MATLAB or Python or wherever, maybe you have experience or you have a team that's working with biostats team that wants to work with it, they are able to take that data and do what they want to it and do whatever treatment they want to do with it. You also get access to the My Metabolon portal, which allows you to map your data onto the pathways that we cover. And then included in that is pathway enrichment. And then as depending on the level of support you need, you can also get some biological interpretation. So that's where one of our translational scientists goes through your data, spends about a week or 2 weeks of their time analyzing your data and looking at the pathway changes and, really pulling on our 20 years of institutional knowledge and all of the experience that we have to be able to help you flesh out that biological story. So they're able to talk about the pathways that are changing and that are relevant to your disease state. They're able to talk about whether that might be a novel change or maybe that's something that is really typically seen, for example, in liver disease or, whatever your wherever your area of research might be. We have a lot of experience. And then they help you just create that roadmap of what's happening and help you determine, where should you go next in your research, and really what's happening at that small molecule level, and really what that phenotype is doing and on that physiological level. We really like to consider ourselves a partner when we're working with you. So we want to be involved as early as study planning. So thinking about how you're going to design your experiment, what's going to be best, how to power your study, how to collect samples. We can help you and talk through all of those considerations. We have 4 different methods that we're running in the lab. So 4 different LCMS methods. We're all mass spec based. So we have different chromatography methods that are utilized to help best capture different classes of metabolites. So we're really covering that and capturing as much as possible. Then we have a whole library and curation team. So they match all of your data to our library, and they curate all of those 4 data files into one data, and they reduce all that data down into that one file, where then we work with it in statistics and do that bioinformatics and that interpretation as well. So in addition to the global panel, we also have, like I mentioned, the complex lipid targeted panel. It's the quantitation of 1,100 lipids with an exact calculation, so an absolute quantitation. Lipids can be really challenging to analyze. Complex lipids have multiple different fatty acid tails. They have different levels of saturation. They can have different head groups. They can have different lengths of tails. And so they can be really complicated and really challenging to analyze. But Metabolon has some good solutions there. So we use an IO mobility-based method to help separate those different classes of lipids, allowing us to be able to look at each different one and categorize the percentages of each of the lipids that we're looking at. We're quantifying all of those lipids. We use 50 internal standards, which allow us to be able to do that. And then we have really good coverage. We are covering 14 different lipid classes of those neutral and nonpolar lipids, and it's exactly what it sounds like. We're really getting those complex lipids and really able to dive deeply into that. Coming along with the complex lipid panel is access to our online portal for Surveyor, where you're able to look at some pathway maps and look at some different statistics and change how you're analyzing your data for that lipid panel, with some nice reference information as well. We have some targeted panels as well that offer absolute quantitation. And I'll just pop up a list here. We have 24 different off-the-shelf methods that are offered. So if there's something more targeted or a more specific hypothesis that you guys are working with, then you're able to utilize one of our off-the-shelf panels and dive a little bit more, take a little bit more of a precision dive into what you're looking at and get that absolute quantitation for each metabolite on the panel for each of the samples that you submit. And I really can't stress this enough. We have a lot of valid or accreditations, which really give you a lot of confidence and know that you're getting the highest level of fidelity of data. Our entire lab is ISO 9001, which means that we have an SOP for everything that's happening to your samples when they come into the lab. So we have an SOP for how we accept your samples and how we're ascensioning your samples and preparing them to how they go on the platform. We have an SOP for how we're collecting the data and how we're curating the data and how we're reporting that data back to you. So you really have a lot of confidence when you're working with us that we're following strict protocols and that you're going to get a really high level of consistent and reproducible data when you work with us. And I'll leave this up for just a moment in case we want to have any questions on the technology or the deliverables. I think Chip is going to pop back on here.
Indeed. Yes. Thank you, Katie. That was awesome. We did have one question about serum volumes for mouse studies. We don't tend to get a lot of volume from mice. Could you comment on that?
Yeah, so we understand that serum from mouse models and some of those smaller animal models can be difficult to collect. We do ask for 150 to 300 microliters as our standard for a biofluid, but we can certainly work with less, and we can have that conversation of what that looks like. We don't like to say no, and we certainly understand some of the constraints that people are working with when they're building out their research and their experiments.
Nice. And then along the lines of tissue or solid samples, can you comment on the amounts required?
Yeah, so it depends a little bit, but it's usually 50 to 100 milligrams of tissue or solid material that we're asking for. Again, if it's, something where you're working with really small models or, maybe you're looking at a small organ, collecting that can be a challenge and we totally understand that. So we can work with less, but then again, that is a conversation that we like to have. So feel free to reach out to Chip or, one of us and we're happy to have that conversation. But 50 to 100 milligrams is usually our limit there.
Nice. And then also a question about sample prep, especially for tissues. Do clients need to do any prep of the samples or just send them in as is?
No, absolutely not. So that is one thing is because of our ISO 9001 certification, we have an extraction method that's approved and that we utilize and that, we work with. And so all we ask is that you dissect your samples and section them how you would like them to be sectioned and send them to us frozen. So you'll flash freeze them and then send us your neat frozen tissue and we'll do the extraction and everything else. So it's really just a dissection and your part in a collection.
Nice. I did see a question about the heavily interpreted deliverable. The question was, do we have an example? Short answer is yes, we do. And we could forward that to anybody that's interested. Last question I had was, is there a path to custom assays if we identify analytes of interest in discovery?
Definitely, definitely. We're happy to do that. Like I said, we like to think of ourselves as being able to help you with all of your small molecule analysis, and that does include custom targeted assays as well. There's a lot of levels of validation and testing that goes into that, but we're definitely happy to work with you on that if you come up with specific molecules that you're particularly interested in, and it's not something that we offer off the shelf.
Nice. Another one popped up. How do you deal with ongoing studies where samples are collected longitudinally? How do you deal with batch effects?
Yeah, so for batch effects, so we do have different ways of normalizing and bridging data. So if you want to bridge two different projects together, let's say you have samples ready to go right now, and then you're going to have samples that are in alignment in the same hypotheses in the same study, maybe 6 months or a year from now, and you want to be able to combine those studies, we certainly have that capability. So we get around some of those batch effects and those normalizations by collecting samples, bridging samples, basically. So there's a few different options for that. If that's something that you're interested in, we're definitely happy to have that conversation again as well. But we do have options for bridging those and, getting around some of that batch day normalization challenges.
Nice. And then the last question is, for cell profiling studies, ballpark, how many cells would be required?
So we ask for 100 microliter packed cell pellet, which I realize is not how any biologist thinks about cells or microbiologist thinks about cells. That's usually in the range of 5 to 10 million cells. Again, we can always work with less. We're happy to have those conversations and give you our best advice and what we've historically seen. But again, that's just a conversation that we're willing to have. So like I said, it's not how people normally or microbiologists normally think about cells, but we're happy to have that discussion and work with you as best we can.
Nice. Thank you, Katie. In the interest of time, we've got a couple of more questions, but maybe we'll move on to the next session regarding the CAR-T application.
Perfect. Yeah, we'll stay on and we'll answer questions after too.
Awesome.
Thanks, Chip. So moving right along, we'll talk about this next case study, the T-cell activation and optimization. So the second case study that I wanted to share with you today is one from a group out of Emory that worked on optimizing T cells. CAR-based therapies can be really effective cancer treatments, but have been less successful against solid tumors. And that can be to a number of different reasons. The main ones really are poor trafficking of those T cells into the tumor. The tumor microenvironment can be overwhelming to the T cells, and some of those malignancies are really just too aggressive. So researchers for a long time have realized that they need to improve the quality of those T cells ex vivo. Literature has shown that the Th17 cells are the most effective, but efficiently expanding those cells has proven really challenging in the past. The activation of these T cells is also very important, and it can impact the functionality of the helper cells. CD3 co-receptor has been shown to be one of the most effective ways to do that. So this is where the researchers focused on this paper. So to get at these questions and to get at this optimization of these T cells, this group worked in human CAR Th17 cells. They were activated to different levels with either the CD28 or the ICOS stimulator bead coatings. So they were able to look at those different levels of activation with the CD3 and either that CD28 or ICOS stimulation. And then they assessed those cells using a combination of flow cytometry, glucose measurements, and then metabolomics, of course, to look at those different cell activation types. And then they also utilized a mouse model with tumors to get at their hypotheses and their questions. The researchers wanted to investigate the theory that expanding the T cells with fewer ICOS or CD28 beads would produce a more effective lymphocyte. And you're able to see here on that flow cytometry, when they lowered the ratio of B to T cells, they had more cytokines that were produced, and they were able to produce a more effective T cell. They also looked at a wide range of ratios. They looked at an effective range. They looked all the way to 1 bead to 130 or 341 cells, but really that effective range was between the 1 to 43 and the 3 to 1 ratios, which were demonstrating that sort of middling range of production, or the highest range of production of those different cytokines that they were examining, and you can see that highlighted there in gray. The memory phenotype is also really crucial to the effectiveness of the T cells. Naive and memory T cells persist longer and exhibit greater anti-tumor activity than effector T cells. So you can see here that the weakly activated ICOS and CD28 cells showed a more naive phenotype. So that weak activation is that 1 to 10 bead to cell ratio, where that strong activation is that traditional way of activating the cells of 3 beads to 1 cell. And the CD45 production, or the CD45 measurement is indicative of a naive T cell, and the CD45R naught is indicative of more of an older or less of a naive cell, as that's indicative of a T cell that's been exposed to different antigens. So you can see here where they were able to make a more homogeneous phenotype when they use that weakly activated cell. They also were able to look at the energetics a little bit and start to get at that for these different T cells. They showed that the weak activated cells took up less glucose from that GLUT1 measurement, which you can see here. So you can see the CD28 strongly activated cells are that black trace. The CD28 weakly activated cells, that 1 to 10, is the blue trace. The ICOS strongly activated is the purple. And then the ICOS weakly activated is the red, and you can see there that they were taking up less glucose by measurement of that GLUT1, and they were also measuring the glucose uptake using that glucose reporter 2-MBDG, and you can see here across the different days that less of that glucose was being taken up by those weakly activated cells compared to the strongly activated cells. So because of these changes in the glucose metabolism that they were able to determine, they wanted to dive a little bit deeper into that and really characterize those changes. So they went with metabolomics. Global metabolomics was performed on these weak or strongly activated cells, and you can see that the strongly activated cells had an increase in glycolysis intermediates. So you can see here on the heat map when we're comparing our strongly activated cells to not activated, or our weakly activated cells to our strongly activated cells, you can see higher amounts of those glycolysis and glucose intermediates. They also analyzed the extracellular supernatant with these different cell cultures, and they were able to show that the weakly activated cells didn't show carbon scavenging and uptake as much, which indicated gluconeogenesis rather than glycolysis in the pathways. They also demonstrated that the ICOS cells had increased levels of lactate, which you can see, which is really indicative of higher levels of glycolytic activity. So taken all together, these data suggest that weakly activated ICOS T cells would be more effective in vivo than strongly activated T cells because they're taking up less carbon from their surroundings. So they're less reliant on that, so the tumor microenvironment would be less harmful to them. So this gave the researchers a lot of confidence that they would be able to utilize these weakly activated cells in treatments with mice with tumor. So they treated in vivo mice, NSG mice that were bearing M108 tumors with their different T cells, with the activated, weakly activated and strongly activated cells. And you can see that the weakly activated cells showed an increased survival, which you can see here on their survival plot. The untreated mice are here in the white trace here. The strongly activated CD28 and ICOS cells shown in the black and the purple traces had less survival rates compared to their weakly activated counterparts of CD28 in the blue trace, and the weakly activated ICOS cells, which survived, those mice survived the entire duration of the experiment. They also had decreased tumor size, which you can see the untreated mice had very large tumors. The strongly activated CD28 and ICOS treated mice had smaller tumors than the controls or the untreated mice, but those weakly activated counterparts, again, had even smaller tumors than their counterparts. And that ICOS weakly activated cell had the even smallest tumors with significant differences between the other treatments. And then again, that weakly activated ICOS group had the most percentage of the CD45, which is indicative of that memory phenotype, that naive memory phenotype. Taken all together, this study really helped validate the energy shift that's happening in those weakly activated ICOS and CD28 cells. Metabolomics really empowered the researchers with the knowledge that the metabolism had changed in these cells and they would be more effective treatments in vivo. And really, this allows for promising research that we can optimize these therapies for treatment and improve solid tumor treatment approaches. And that one was pretty short and sweet, but what can Metabolon reveal for you? Thank you so much for coming. We're happy to answer questions. I'll let Chip jump back on here and moderate any other additional questions that we have.
Thank you for that, Katie. We had one question about CAR-T applications for or cell therapies grown in culture, where Metabolon has been used to troubleshoot or optimize growth conditions and yields. Is that a growing common application that you see?
Yeah, for sure. You know, getting those cells optimized is something that, you know, researchers are struggling with. Like we've seen, you know, getting those cells to proliferate and you know, expand ex vivo is a challenge. And so being able to dial in their metabolism is something that is really impactful to that research. And that's definitely somewhere where Metabolomics can help you and where we've obviously done a little bit of work as well.
Nice. And then for folks that are doing studies in that realm, how do you get around or address media contributions? Are there any special considerations for subtracting media background?
So we analyze media pretty routinely when we're looking at cell culture and cell culture studies. And so that's something that we're happy to do. And that can be really impactful when you're looking at, you know, what the cell is taking up, like that last case study that we talked about. And that is something that what we do is we just ask for fresh and spent media when we're doing those analyses. So we'd ask for a fresh sample of your media and then all the different spent medias at your different time points or your different treatments.
Nice. I think that's all the questions that we have. We'll just give it another second to see if any others pop up.
Yeah.
As we're waiting for that, I just wanted to let the folks online know. So the 3,000 publications that Katie referenced, there's a lot of Nature papers in there. And especially for the gene therapy folks, there's a good number that have involved UK Biobank studies, FinGen, ERIC, TopMed. So if there's any interest for some of those links to those papers, we're happy to forward those to you as well. So if interested, just let us know. I don't see any other questions. So if there are no other, I guess we'll adjourn. Just wanted to thank you, Katie, for your time and thank everybody for attending today. We will have a recording of this webinar available in the next couple of weeks. So if that too is of interest, just let us know and we can forward you that as soon as it's available.
Great. Thanks so much, Chip. Thanks everyone for
Very
listening.
good. Thanks for your time.